I'm so honoured and grateful to be the recipient of the Rowley Prize this year, and I thank the International CML Foundation for recognising my team's contributions. BCR-ABL is at the heart of CML, and its behaviour influences both response and resistance, and my work overlaps both. We studied milestone BCR-ABL values, transcript kinetics, and deep molecular response to predict outcome and treatment-free remission, and the mechanisms of drug resistance, including BCR-ABL kinase domain mutations and other mutations in blood cancer-related genes. And we've recently detected additional rearrangements that occur at the genesis of the Philadelphia chromosome. But I'd first like to thank and acknowledge my molecular mentors, Barney Rutsky and Tim Hughes, who set me on this journey when they took me on as a technician to develop a method to quantitate BCR-ABL, and then became my PhD supervisors. So I'm really grateful for their help and support along the way. Now, long before we used the BCR-ABL International Reporting Scale, I was fortunate enough to work on clinical trials with these pioneers of CML PCR testing, where we perform molecular analysis as an exploratory test for patients enrolled in the first clinical trial of imatinib. And this was the IRIS trial. And among the findings from this study was the prognostic significance of achieving early milestone BCR-ABL values. Initially, we used a log reduction scale, and in a sub-study, we found that a 2-log reduction at 3 and 6 months of first-line imatinib was predictive of outcome, and this included treatment failure. But it was a decade later, after standardization of molecular methods and the introduction of the international scale, that the European Leukemia Net first introduced molecular milestone values for treatment decisions, and this was followed by the US National Comprehensive Cancer Network. But there was debate about the milestone value of 10% at 3 months and whether to proactively change treatment to avoid the risk of disease progression. So we and others did further work to refine outcome prediction based on molecular values and found that the rate of decline was a better predictor of outcome than a single value. So shown here are 2 patients with BCR-ABL values in the warning category at 3 months of greater than 10%. So these patients are at risk of treatment failure, but we don't know if one is at greater risk. So we compared the 3-month value to the individual baseline value. And patient 1 has clearly had a response, whereas patient 2 had no response. And the downward trend continued for patient 1, and patient 2 failed therapy. Now we assess the rate of decline as the halving time, where a shorter halving time indicates a more rapid decline. And both the ELN and NCCN suggest that an assessment of the kinetics of response can aid treatment decisions. And importantly, a number of studies have shown that quantitative PCR methodological differences don't preclude the broad adoption of the assessment of the initial kinetics of response for outcome prediction. And in work published this year by my PhD student, Narini Shanmuganathan, we also found that the early BCR-ABL kinetics predict treatment-free remission. 2 patients are shown who had a similar BCR-ABL value at 3 months, but patient 1 had a more rapid decline than patient 2. And those with the most rapid decline had a really high rate of treatment-free remission of 80%, and those with the slowest decline had a very low rate of TFR at only 4%. And we thank our friend and colleague Dongwook Kim from Seoul St. Mary's Hospital for providing a number of validation cohorts for our studies. So the early response kinetics are predictive of both short and long-term outcomes and may reflect intrinsic biological differences between patients. And Narini is also working on mechanisms of TKI resistance. And we've had a major interest in studying these mechanisms, and we're among the early groups who identified BCR-ABL kinase domain mutations and their clinical association with resistance. And this work was made possible by the support of Novartis, in particular Dr. Kevin Lynch, who was the Oncology Medical Director for Australia. And he's shown here in Sydney earlier this year with Junior Malo, who was a pioneer of imatinib resistance research and a friend now living in Adelaide. So Novartis were very keen to understand the causes of resistance so they could improve therapy. And I'd also like to acknowledge a number of colleagues that we've worked with over many years, and a postdoc in my lab, Wendy Parker, who developed a mass array method to detect low-level BCR-ABL kinase domain mutations. And she was the first to show an association between low-level mutations and outcome, as well as the prognostic significance of having multiple BCR-ABL mutations. Now, we, like many other groups, found lots of mutations within the kinase domain, and among the most common were those located in the phosphate binding loop, the P loop, that are coloured here in blue. We found a very strong association with poor survival for patients with P loop mutations, and this finding was confirmed by some, but not all subsequent studies. But our clinical observation was confirmed by Michael Deininger's group, where they compared the transformation potency of 5 common kinase domain mutations that are shown in red, and they used various biological assays. The P-loop mutations Y253H and E255K exhibited increased transformation potency relative to wild-type BCR-ABL, which was consistent with altered substrate specificity and pathway activation. M351T and H396P were less potent, and T315I was assay dependent. So the work confirmed that the transformation potency of mutants differed, and it's this that may determine patient outcome. And kinase domain mutation analysis continues to be an integral component of patient management. But over the last few years, evidence has accumulated that mutation of cancer-related genes other than BCR-ABL is associated with treatment failure in CML, including primary resistance. And a review of the literature on behalf of the ICMLF Genomics Alliance found 15 studies that reported mutations in these blood cancer-related genes in patients at diagnosis. And in our study, we performed whole exome sequencing, RNA sequencing, and copy number variation for 46 patients at diagnosis who were selected based on either having a good or a poor outcome. And we found a significantly higher frequency of cancer gene mutations in the patients with a poor outcome. We performed the same analysis for 39 patients in BLAST crisis and identified multiple mutation types. Most frequently, frameshift, missense, nonsense, and splice site in 72%. But we also detected cancer gene fusions in 31% and cancer gene deletions in 36%. So by combining the different mutation types, we found that 100% of patients in BLAST crisis had one or more mutated blood cancer-related gene. And 58% of patients had BCR-ABL kinase domain mutations, and every one of these patients had additional mutations in cancer-related genes. So BCR-ABL dependent and independent resistance mechanisms frequently co-occur. But we also detected novel fusions and rearrangements in 24% of patients at diagnosis. And these were fusions of genes on chromosomes 9 and 22, or fusions that correlated with variant Philadelphia chromosome translocations. Most involved BCR or ABL, and some were associated with deletions. The majority were complex and involved inversions and novel circular RNA. We even found evidence of sequence fragmentation and random reassembly that generated novel fusions. And we've termed these Philadelphia-associated rearrangements and believe they occur at the time of formation of the Philadelphia. Now, work done by others in the pre-imatinib era reported deletions of the derivative 9 detected by FISH analysis that were an independent prognostic indicator in CML. They were detected in 10 to 15% of patients and occurred at the time of formation, again at the Philadelphia chromosome. But in the TKI era, these deletions don't have prognostic significance. But the Philadelphia-associated rearrangements that we've described using next-generation sequencing extend well beyond deletions. And I'll illustrate what we found by first showing the usual events that generate the Philadelphia chromosome. Shown here are the locations of BCR and ABL on their respective chromosomes. The translocation generates the Philadelphia and the derivative 9. And we've detected the reciprocal ABL-BCR fusion in about 60% of patients. And here is an example of a relatively simple Philadelphia-associated rearrangement where a novel reciprocal fusion was identified, a USP20 SPEC1L fusion. USP20 is located upstream of ABL and SPEC1L is downstream of BCR. Copy number analysis revealed 2 approximately 1 megabase deletions that are shown here in green. And the deletion breakpoints occurred within these 4 genes. Now we speculate that the translocation generated the standard BCR-ABL fusion and the novel reciprocal fusion on the derivative 9 due to the deletions. And this patient had a very poor outcome. Lymphoid blast crisis at 6 months of first-line imatinib, a P-loop mutation, and additional chromosomal abnormalities. And whether this novel reciprocal fusion is a driver lesion is unknown. But work by others has shown that USP20 is a deubiquitinating enzyme that may function in genome maintenance and DNA repair. So it's a possible tumor suppressor. And the Philadelphia-associated rearrangements were enriched in patients with a poor outcome. And here is a summary of the genes and their location on chromosome 9 and 22 that were fusion partners in our cohort. The green text indicates the genes involved in inversions, and these occurred in 82% of patients with a Philadelphia-associated rearrangement. And the green bars indicate deletions and their relative size, and these were detected in 9% of all patients. Now, some of the deletions were non-contiguous, and these generated highly complex events. And that's illustrated in this example. Now, each of these genes were fusion partners, and 5 non-contiguous deletions aligned with the fusions. The standard BCR-ABL fusion occurred. From the position of the genes on their respective chromosomes, the expected orientation of these reciprocal fusions is chromosome 9 genes in orange as the 5' partner, and chromosome 22 genes in blue as the 3' partner. But we found the opposite, and some of the genes were in the inverted orientation. So we postulate that this could only occur if there was sequence fragmentation, inversion, and random reassembly generating novel fusions that were cytogenetically cryptic. Now, this patient achieved an optimal response at 3 months of first-line imatinib. And among the fusion partners was NEC6 that has a cell cycle-related function. And work by others has shown that interference with NEC6 function induced a marked growth inhibition, mitotic arrest, and apoptosis in cancer cell lines. And the TNR-C6b NEC6 fusion was out of frame. And it raises the question whether loss of NEC6 could function or could enhance the apoptotic response to imatinib. And this is similar to some BCR-ABL kinase domain mutations with less transforming potency compared to wild type. Now, in new work, we've now sequenced a validation cohort of 202 consecutive patients treated with imatinib using an RNA-based gene panel and identified 19% with a Philadelphia-associated rearrangement. And the complexity of these rearrangements is shown in this circos plot that was generated by Narani. The central colours indicate either direct fusions, inversions, or circular RNA. And we have found an association with poorer outcome in this validation cohort. And this work was presented by Narani at ASH last year. And in particular, we found a very strong association with the subsequent acquisition of BCR-ABL kinase domain mutations. Now, each one of these rearrangements were unique to individual patients, and they may be a marker of genomic instability and raises the question whether leukemic cells with Philadelphia-associated rearrangements have a predisposition to accumulate additional genomic damage. So the take-home messages from our studies: the initial kinetics of BCR-ABL decline after commencing TKI therapy provides short and long-term predictions of outcome, including future chance of treatment-free remission. BCR-ABL dependent, that is kinase domain mutations, and independent blood cancer-related gene mutations frequently co-occur. And new technology has revealed that the formation of the Philadelphia chromosome is more complex than once thought, and this complexity may contribute to outcome. So in addition to the people I've already mentioned, I'd like to acknowledge the people listed here, including the staff from Genetics and Molecular Pathology at SA Pathology, our bioinformaticians and the genomics facility staff, the people at SAMRI, our funding bodies, including the pharma industry, and Cepheid, our collaborators, and particularly our patients and the CML Advocates Network. Thank you.